Hello, everyone, and welcome to today's live broadcast, Reproducibility of Animal Studies and Animal Welfare Ethical Perspectives. My name is Jane Pham from LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots, the leading scientific social networking website and producer of educational virtual events and webinars. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would like now to introduce our today's speakers, Stacy Pritt and Vatsala Naageshwaran. Stacy Pritt is the director of the IACUC office at the University of Texas Southwestern Medical Center in Dallas, Texas. In this capacity, she oversees regulatory compliance for one of the largest animal research programs in the United States. Dr. Pritt holds a Bachelor of Science degree in biology from the California State Polytechnic University at Pomona and a Doctorate of Veterinary Medicine degree from Washington State University. Dr. Pritt is a nationally recognized authority on regulatory compliance, including IACUC operations, adherence to federal regulations and policies, maintenance of good laboratory practice procedures as dictated by the U.S. FDA. Vatsala Naageshwaran is currently the Director of Operations at Absorption Systems San Diego and oversees scientific and business operations for the company. Previously, as the Associate Director of Scientific Operations, Vatsala collaboratively drove research and expansion of cell port absorption systems, exclusive drug transporter technology. She's on the Scientific Advisory Committee of Society for Pharmaceutical Dissolution Sciences, SPDS. In her current role, Vatsala also helps support absorption systems multi-platform testing, integrating the use of in vitro, ex vivo, and in vivo models, for testing NCEs, devices, novel drug delivery systems, regenerative and gene therapies, formulation performance assessments, and augmenting equivalents of generic products. I will now turn it over to Stacy for her presentation first, followed by Vatsala's presentation later.
Thank you for that introduction. I'm very excited today to speak to this audience about reproducibility of animal studies, specifically animal welfare and ethical perspectives on this topic. I have about 24 slides, and I am actually going to use the polling function as well, which I'll talk about in a few seconds. So as far as the overview of what I'll be covering in my part of the presentation, I've got a few words to say about the purpose of why are we here and why are we talking about reproducibility. And with that, I do actually have several graphics, comics in the presentation. There's a lot of stuff out there on the internet about reproducibility, so I thought I would share some of these sentiments. So this one reads, are taxpayers getting their money's worth with the research? And I'll assume it's a PhD advisor. He goes, that's a dangerous question to ask. It's called research for a reason. We're at the cutting edge. We can't guarantee results. And then most likely this PhD student is asking, I don't have to guarantee results. And the advisor says, only to me. I am going to talk briefly about ethics, and I'm going to spend a little bit more time on animal welfare. And I want to talk specifically about IACUC responsibilities in this whole arena of reproducibility. With my polling questions, I do want lots of audience participation, and then I have a few conclusions. So what's our purpose here today? One of the cornerstones of the scientific endeavor is actually to be able to learn from the research of others. That's very important with how our current scientific infrastructure is has been built and how it's maintained and how it will be advanced. This relies on the ability to repeat studies. This is part of what's called validation, validating of animal models. We know that this animal model is a model for this disease, for example, and it helps us to apply new treatments and new therapies. Researchers really build upon previous studies. They definitely look to repeat certain elements or components of previous studies to make their research better. And one of the cores of animal research is to be able to apply the information to human research or to be able to predict outcomes in human research. And I did want to put a little bit of a disclaimer on this talk. For purposes of my session, I'm actually going to be U.S.-centric, and I'm going to be talking about U.S. guidelines. However, as I'll state here in a few slides, when you start talking about animal welfare and ethics, many times you're talking more about questions and sometimes questions that don't have answers. And so a lot of the questions that I pose can be applied regardless of which guidelines I'm applying, U.S. or otherwise. So in 2014, ILAR, the Institute for Laboratory Animal Research, held a roundtable talking about reproducibility. Some of you who have attended other webinars and other sessions within this virtual conference have probably seen references to this roundtable. Within the report from this roundtable is a statement. It talks about recent publications and statements demonstrate concern regarding the, quote, prevalence in the number of peer-reviewed studies that cannot be reproduced, particularly those containing data from experiments using animals and animal models. When we don't have reproducibility, when we don't have the ability to validate an animal model, when we don't have the ability to apply our research to human research or predict outcomes, this endangers the entire scientific endeavor. It can erode the integrity of and public trust in science. And repercussions down the road, we could have decreased funding and support for science, for scientific research, and we could have less translational research. So again, less research going into humans. A lot of pharmaceutical companies are discussing their inability to reproduce studies. And a lot of pharmaceutical companies for many years now have increasingly turned towards academia for the discovery of new molecular entities. The pharmaceutical company develops those new molecular entities, and then you get new drugs and treatments. So if they're not able to reproduce those initial studies, that's a problem for our drug pipeline. So I'm going to do a polling question. So the question is have you heard about reproducibility issues in animal research before? And I mean before this virtual conference. So far everybody has. And we've got about a fourth of the attendees responding. Oh, a couple people have now said no. I'm going to push these results here in a second. Okay, well, I'm going to go ahead and push the results with 27 people responding. So the vast majority of people have heard about reproducibility issues before. So what are some of the causes of this lack of reproducibility that people are talking about? The number one cause that I see, especially within the scientific versus the lab animal or regulatory literature, is statistical underpowering. And that's simply not having enough animals to make sure that you're picking up a statistically significant result. I have a little bit more information about this in some later slides. There's the incorrect interpretation of data, and a few authors have attributed some of this incorrect interpretation to unforeseen technical issues, which have resulted in a lack of reproducibility. There's a discussion about researchers not appropriately describing control issues and or contradictory or even all of their data. How much research, how many other groups, how many other things were studied before that particular scientist published the data set that they published. Then there's a couple concepts around this self-correction and correcting the scientific record. There's a feeling that within our current scientific construct that we're not going about correcting the scientific record very well. So when researchers find that there's non-reproducible data or there's incorrect interpretations that are detected, have the appropriate retractions been done? And so for many years, science has relied on this self-correction within the scientific record, but that seems maybe to not be working now. And the problem with that is that papers with fundamental flaws then live on, and I'm quoting from another source there. But that means papers that can't be reproduced end up getting cited, and that causes problems. These concerns are both with actually animal and human research. And a little bit more upon the statistical underpowering, that's actually occurred especially in neuroscience. There's actually been some publications in that field. And as I mentioned, underpowered studies mean that not enough animals were included such that true statistical differences can be detected. What happens when you have underpowered studies is there's an increase in false positives. So the positives that you see are actually more likely to be due to chance, so a false positive, than be due to anything that's experimental. And with the incorrect interpretation of data, that's where concern with fraud and data manipulation can also come into play. There's this one reproducibility project that I saw. Actually, a researcher from my institution is involved with this. And this particular project, the HHMI, Howard Hughes Medical Institute, is involved with it, is looking to try to replicate 50 pivotal research projects in cancer biology. It started in late 2014. They've only worked themselves through a few projects, but this kind of work is currently ongoing. So my final comments about our purpose and why are we here. I am going to review the basic concepts of ethics, animal welfare, and IACUC responsibilities as they relate to reproducibility. There's a lot of questions. I'm going to be putting forth a lot of questions that's inherent when you start talking about ethics and animal welfare. I've got a few scenarios that we can discuss, and just another little cartoon. One researcher says to another, there's a fundamental flaw in your experimental design. All the mice are Scorpios. And there's a few potential answers that we're going to examine, but there haven't been that many answers given in this field. So moving on, we're going to start with ethics. There are many ethical theories out there, just in general. The study of ethics I find to be personally fascinating. The need for animal research is actually usually justified from the quote-unquote greater good concept, which stems from an ethical theory called utilitarianism. Within that theory states that the most moral action is the one with the best outcome. So when you talk about a moral action, if your moral action is to utilize animals in research for the benefit of human and or animal health, then that's the best outcome and you should do it. And that greater good is that animal research benefits both animals and humans. I'm not using the 3 R's as an ethical theory. The 3 R's serve as a theory for, or serve as a foundation for for a more ethical use of animals, but they don't serve as an ethical basis for the absolute use of animals in animal research. And for those of you not familiar with the 3 R's, briefly, it's the words themselves are replace, reduce, and refine. Replace animal models wherever possible. Reduce the use of animals, although that's not quite the correct interpretation, and then refine the work of your work with animals. It's not my purpose today to go over them, but I wanted to put them in context with some of my later comments. So again, the 3 R's provide structure for investigators using animals. They are incorporated into some regulatory guidelines, and I'm going to discuss a little bit more when we talk about animal welfare. There's a core concept when it comes to discussing ethical issues and reproducibility. The core concept is studies need to provide specific or universal information that can help with other research in order to conform to that greater good ethical argument for animal research. Studies have to provide that information. As we saw, a cornerstone of science is to build upon the research of others. But if a study is not reproducible, therefore it probably is not contributing to that cornerstone of science, learning from others. Is it therefore ethical? So if the study is not reproducible, is it therefore ethical to perform? So again, another little cartoon. One researcher is calling another researcher. I followed the methods in your paper to the last detail. The experiment doesn't work. The other researcher says, well, when you did the experiment, when did you do the experiment? On a Friday? Oh, that's why. It never works on Fridays. This one, in a very pointed way, describes some of the whimsy that may go into why something works and it didn't work for somebody else. So what you have when you ask this ethical question, if the study is not reproducible, is it ethical? When you don't have reproducible animal studies, other than some of the other things I talked about as far as eroding trust in science, people start saying that animal lives are now wasted. We have wasted animal lives. We have wasted money and time. And we've put now the safety of clinical trial subjects, so that's people enrolled in human research, we have now caused safety problems for them. So there's huge ethical implications for not having reproducible animal studies. So polling question number 2, what do you think? If a study is not reproducible, is it ethical? There's no right answer to this. There's many times not a right answer with ethical questions, but it's great to get the audience involved. And I did give you the option of saying depends if you don't really want to commit. Okay, we have about 23 respondents, and 8 of you said no, and now 17 of you said it depends. So I wanted to point out 3 areas of the U.S. regulations for animal research that could relate to this area of reproducibility. I wanted to go over the concept, though, of validity versus reproducibility. Validity in the statistical concept, and I say this without being a statistician. So validity is the conclusion or measurement that is well-founded and it corresponds to the real world. It's the degree to which the conclusions about the data are correct. Reproducibility, though, is really more about the replication of statistically significant results. In my world, you could have a valid study results. Those results are valid for that particular data set examined, but that doesn't mean that that particular research can be replicated or reproduced. You would most likely need validity, though, in order to have reproducibility. So then we take a look at the U.S. regulations. The USDA states that IACUC approval requires that the PI is provided written assurance that activities do not unnecessarily duplicate previous experiments. The number 3 U.S. government principle for the utilization and care of vertebrate animals states that the animal selected for procedure should be the minimum number required to obtain valid results. And OLA says that scientific justification should be utilized as much as possible for experimental and control groups. Also, in other areas, OLA talks about minimizing the number of animals used. So therefore, we do run into some questions when we even look at our own regulations. Does performing work for the sake of reproducibility then constitute duplication of previous experiments? Who's going to make that decision? Is that going to be the IACUC? Is that going to be the USDA? Should PIs aim for reproducibility when the U.S. government principles actually desire validity? And those are two different concepts. So very important questions to ask. Moving on to animal welfare. Brief definitions of animal welfare. Animal welfare as a science started in the late 1960s and early 1970s. Around in that early mid-1970s, the 5 freedoms for animals came about, and that started in England. And I'm not going to go into it, but at this point in time, these 5 freedoms are considered kind of a baseline for animal welfare and the areas for assessment when you're professionally assessing animal welfare, in particular farm animal welfare. There's also the thought of coping. This is by Broom. And Broom states that the welfare of an individual is its state in regards to attempting to cope with the environment. And an animal with good welfare is the one successfully coping with the demands of its environment. The definition that I like for animal welfare is a more current definition proposed by Fraser, and this is where you look at 3 different aspects of the animal: its health, how it's able to perform normal behaviors and its emotions, that's the affective states, as well as elements of natural living. So the point here that I want to make is animal welfare is not animal health. Animal health is actually, in its broadest terms, a part of animal welfare, but animal welfare includes more. So with animal welfare and reproducibility, that's a little less defined, and people really haven't spoken about it as much, in my opinion. The questions that you have with this are, how is reproducibility ensured in different research environments with varying levels of animal welfare? That one I've seen alluded to. This next question I have not seen discussed. Could or should compromises in animal welfare make the model more or less reproducible? I have some detail on this. So when you start looking at ensuring reproducibility in different research environments with varying levels of animal welfare, what are we talking about? We're talking about animal housing, animal health, the disease status or genetic manipulations of the animals, animal behavior, animal affective states, their emotions. Some other things, non-animal related variables that appear in the literature, is people using different equipment, even different reagents can make a difference on the reproducibility of the research. Some of the answers people have discussed in order to address this particular topic have been that we need to publish the details. And indeed, some journals are asking for more information, which is probably a good thing. There has been some discussion about asking for a group, whatever that group may be, to establish more detailed standards. That thought, though, has not gained traction because there is the concern of increased regulatory burden with that. So in talking about compromises in animal welfare, should you have a compromise in animal welfare to make the model more or less reproducible? What are we talking about? So here's a scenario, and this is where a PI is proposing to perform a study with a standard animal model. However, that standard model actually calls for different animal-related practices than what your institution has established. and you feel in your institution that your standards are giving you a very high level of animal welfare, do you lower or change your standards for the sake of animal study reproducibility? Again, there's no right or wrong answer here, but this is the animal welfare part of reproducibility. So this really provides a natural segue into talking about IACUC responsibilities. And again, that 2014 workshop from ILAR had some very specific points about what the IACUC should be doing in terms of reviewing protocols and oversight of research to help try to ensure reproducibility. Although that is not really within the realm of responsibilities for IACUCs if you look at in the United States at our regulatory framework. So this workshop talked about how the IACUC certainly has a responsibility to look at the humane handling, care, and treatment of research animals. They also recommended that the IACUC do look at scientific aspects of protocols that will affect the welfare of laboratory animals. And I went through some of those animal welfare components earlier. And this also talked about how hypotheses are being tested, what the sample sizes are, and adequacy of controls. So some questions for IACUCs then with reproducibility. Should IACUCs allow or approve work that they know may not be able to be reproduced? You're asking, well, how could this happen? Well, you could have a PI wishing to replicate or slightly modify a study that's published. It was performed at another institution, but that IACUC actually allowed something that your IACUC doesn't. A wonderful example of this is tumor policies. A lot of institutions have a tumor policy saying that the tumor can't go above a particular size. That might be 2 centimeters. It might be 1.5 centimeters. There are a few institutions that don't even follow sizes anymore. They have other criteria. That actually brings up obvious questions on how those studies become reproducible. But if your IACUC says, this is our tumor evaluation method, and another PI says, well, in order to build upon this work, I need to have different criteria, that really puts the IACUC in a bind. And so that's certainly something that could happen. I've actually also seen the second scenario, where results have been published, but then I've either found the retractions or I've talked to that institution's IACUC and found out that there's been compliance issues. What do you do then when your PI wants to reproduce those studies? Brings up some very interesting questions. And so this will be my third polling question. Should IACUCs allow or approve work that they know may not be able to be reproduced? Oh, and I think I pushed a button too soon. Okay, there we go. So again, I have yes, no, and not sure. Well, great participation here. I'm going to push out the results. 56% said no, 28% said yes, and 16% said not sure. So moving on with this, now we start getting back to one of those concepts in the 3 R's of reducing the number of animals used. That may not be the correct interpretation of the 3 R's, in that seminal paper from 1959 actually referred more to reducing animals that are going to experience unalleviated pain and distress. But nonetheless, many people have interpreted that to reduce the number of animals used. So we have a regulatory framework for that in the United States and in other countries, and many IACUCs adopt this. So therefore, should IACUCs then ask that animal numbers actually be increased if IACUC review reveals inadequate animal numbers for hypothesis testing and sample size? So we have a group saying IACUCs really should look at hypothesis size. control group, sample size. Because if you don't have enough animals, the study's not reproducible, but you have the 3 R's saying maybe we should minimize animals. Those concepts can be rather conflicting at times. So here's some of the scenarios I've seen with this. You have a sample size that is too small to derive statistically relevant conclusions. I am lucky in that I do have a biostatistician on my IACUC, and this is very helpful to ascertain. You can see in study design that you might have inadequate or non-existent positive and negative control groups, unaccounted for sex and strain differences, as well as unaccounted attrition. If you're going to do a 2-year carcinogenicity study in rats or mice, you're going to lose some naturally just due to natural lifespan limitations. Has that even been accounted for in your animal numbers? And as I mentioned before, underpowered studies do lead to more false positive results, causing problems with reproducibility. So another polling question. A little bit different here. Has your IACUC ever asked a PI to consider increasing animal numbers, either in a total or a group, in order to make the study more valid? Remember, I'm saying valid here, not reproducible, because we do have a responsibility to make sure that the studies are valid. And I'll add my answer to this. At my IACUC, yes, we have asked for more animals to help improve validity. So great response. 46% said yes, 20% said no, and 34%, now 36%, said not sure. So this little cartoon says, with this much grant money, the only experiment we can do is flip a coin. So I have a quote here. It's something on their website from the American Physiological Society's Reproducibility Journal Club. So their question is, do animal care and use committees and ethical principles such as reduction, replacement, refinement, the 3 R's, create pressure on investigators to propose underpowered studies? This could either be within their grants or within IACUC protocols. So another way of thinking about this is, do the 3 R's and other IACUC or regulatory mandates, in addition to cost and lack of statistical understanding, which are also things discussed quite a bit in the literature, contribute to those underpowered studies that we discussed before? So last polling question here. I want to know your opinion on this. Do animal care and use committees and ethical principles such as the 3 R's create pressure on investigators to propose underpowered studies? And I have a not sure there for folks who don't want to commit. Okay, so I'm going to go ahead and push the results. So we had about 50% saying yes, they agreed, or thought that that did happen. 37% said no, and 11% said not sure. So with this, I'm going to conclude. We're going to go switch over to Vatsala's presentation, then we'll have Q&A. For my conclusions, some kind of take-home points here is that in this area, when you start talking about ethics and animal welfare and how it relates to reproducibility of animal studies, there's a lot more questions than there are answers. And a lot of this is subjective, and a lot of it takes into account like a judgment call as well as institutional culture. One thing I wanted to make sure in people leaving this webinar is that they have an understanding, albeit very superficially, that IACUC policies and reviews can actually impact reproducibility. I've certainly seen it several times since being in a large academic institution. And that the regulations specify validity, not reproducibility. So when we're talking about reproducibility, we're talking about something outside of our regulatory framework, at least here in the United States. So with that, I'm going to turn it over to Vatsala. Thank you.
Okay. We heard Stacy give a great overview just now and raise some really important questions on ethics and concerns with using animal models that are not reproducible. Well, my presentation will focus on case studies and examples that I hope will highlight the sensitivity and reproducibility of using intelligently designed animal models to help predict human outcomes. But I'd like to start by thanking Stacy and LabRoots for inviting me and giving me this opportunity to participate in this very nice seminar, this webinar. And my name is Vatsala Naageshwaran. I work for Absorption Systems, a company that supports testing for innovator and generic product development. So let's bring the in cerebro into the in vivo, as a reviewer recently eloquently put it. I mean, it all goes to intelligent design. So before, too often we rush into a study, I think, with a standard design, trying to replicate a paper without looking at some of those important factors like statistical power, the choice of animal model, the importance of data interpretation. But undoubtedly, there's no question that preclinical models have tremendous value and play a critical role in establishing safety and efficacy within the context of a living model. So as all of you are aware, drug and device testing begins with extensive lab research, which can involve years of experiments that use in vitro, ex vivo, and ultimately intact animal models. And if this research is successful, you get to send your data to the FDA for approval to continue research and testing in humans. So this research that involves lab animals, which we shouldn't forget, leads to many medical breakthroughs, improving lives and providing additional improvements in diagnosis, treatments, and cures. So an important aspect also to establish is the complexity of these animal models. When we look at it, you know, we see the similarities and differences, and too often we're focusing on similarities when we think of predictive or translatable models. But it's important to emphasize that these robust, complex models, that there's value in similarities and differences, because it gives us insight into complex human biological systems. So as a preclinical CRO, I think we have a great advantage here, because we have the ability to work with drugs that belong to different structural, therapeutic, and chemical classes, devices that are being developed for a variety of disease indications and therapeutic endpoints. So this really enables us to work with a very broad spectrum of animal models, small and large, within a highly regulated environment that includes oversights by bodies such as our own IACUC, of course, and then the USDA and FDA and ALAC. And what we have the ability to do is to support those initial sort of non-GLP studies to test efficacy, to look at pharmacokinetic parameters, and support that iterative process of developing a new drug or device, and leading up to ultimately that the safety studies that are conducted under GLP conditions, which provide the basis for approval for testing in humans. So everything we do really is driven by these important outcomes, which is that you're trying to enable a successful IND or NDA submission if you're a drug company, or a PMA if you're a device company, or these models could be utilized to enhance product value, the so-called 505(b)(2) route or the 510(k), where an existing therapy or device is being modified for a new indication or dose route. And finally, the ANDA route, which is used for the development of generics. And I'll talk a little bit more about this last one towards the end of my slide, which is a very important area where the use of animal models to biocharacterize a generic product to demonstrate equivalence is becoming particularly important. So, we have the classic preclinical models here, which are used for efficacy, PK, and safety studies. These include rodents, rats and mice, the rabbits, canines, swine and ovine model, and of course the non-human primate. And I'm not going to really have examples to speak because this is a model that we have less experience with, but we'll have a lot of other great case studies and examples highlighting the utilization of some of the other models. So in the coming slides, what I'm going to really cover will be the development of different products based on dose route or indication that will highlight the benefits of utilizing reproducible and robust and predictive preclinical models. And we'll start with, the obvious place, which is the development of oral products, which is, always a gold standard. All companies want to have an oral product. From a patient perspective, no one wants to get an injection. They'd much rather swallow a pill. So this is a very classic model, very routinely employed, whether it's a rodent or a larger animal like a dog, where you are trying to obtain PK parameters. You're looking at absorption, you're looking at distribution, metabolism, excretion, and of course it's also the PK-PD correlation, which is very critical because you're looking at the exposure associated with compound administration and target engagement. So very commonly, dose routes are evaluated against a parenteral route like IV to get these parameters like bioavailability or Cmax, Tmax, your half-life, volume of distribution, clearance, and so on. Very critical to have those supplementary aspects to this, which I'll briefly touch on, which is the development of an appropriate formulation, taking into account excipients that can be tolerated within this particular animal model. And this really goes to the experience of the lab that is conducting these studies where they have observed the impact of excipients on a particular animal model, if it can have an adverse effect, which can confound the results. So this is very important to keep in mind. Also bioanalytical consideration. So LC-MS is, of course, a very common platform for small molecules. But it's very important, again, in the context of animal models to think about things like matrix effect. If you're analyzing plasma samples, you perhaps might want to consider taking the matrix for your calibration standards from the same animals and so on. All these go to highlight the point and question that Stacy had also raised about the importance of designing the animal study, taking all these different critical factors into question. So these are sort of, routine studies that are performed where you're using ideally the same group of animals that are washed out appropriately, and then, taking blood samples over a time course to obtain these different parameters. However, more complex models can be derived using these very same animal models. For example, you could do endoscopic dosing in dogs where you can observe the impact of regional absorption. Is there a component of site-specific absorption? Is there gastric instability? And we can further extend this out to a surgically modified model where you can port a rat or a dog's gut with intestinal ports, again, giving you the ability to directly dose to a target site in the gut, and then, maybe enable development of a sustained release formulation, for example. So going from simple to complex and the more mechanistic aspects of using some of the animal models, here's a great example because, food, as we all know, can impact pharmacokinetics of a drug substance through a variety of mechanisms. It can improve drug solubility, you can have increased GI motility, you can have delayed gastric emptying, and so on and so forth. And those are several in vitro techniques like dissolution and biorelevant media that have been successfully utilized to predict human plasma profiles after oral administration in the fed state. You need to have a proper formulation for these dissolution testing, which is usually not available earlier in development. So you don't have a clinical formulation at that point. So how are you going to be able to predict if there's a food effect? So the dog is very well studied for understanding food effects. We've established this model. And you can really explore the mechanism as we have done in this particular example with an oncology drug product where we're comparing a nanoparticle suspension to a reference product under both the fed and fasted state. And there's a lot of ways you can manipulate and refine this model. You can use pentagastrin or famotidine pretreatment to influence gastric pH. There are other treatments to inhibit bile flow and so on. So there's really a tremendous value to this model to be able to understand or be able to predict something critical like food effect for an oral drug. Extending beyond oral is the fact that many of these animal models, especially the canine model, is very suitable for evaluating alternate routes of administration. What I show you here is an example of intranasal exposure comparison with oral exposure, because intranasal is very important as a route of administration. It has a lot of importance in an ER setting, for example. The clinicians have shown that naloxone, for example, has a bioavailability of 100% after intranasal administration. So it's as effective as IV, and so there's a lot of development in this space, and the nasal mucosa is richly supplied with blood vessels. It can, result in very rapid absorption and also avoid things like hepatic first pass and so on. So we often see these types of requests to evaluate formulations by these types of alternate routes and compare exposure to see if there is a benefit in developing this type of formulation. Intraoral is yet another area which is becoming, very, very critical in terms of the importance of, the types of products that can be developed using this particular dose route. And when we look at the preclinical models, there's a nice comparison here of the oral mucosa tissue between humans, pigs, dogs, and rabbits. And in terms of the thickness of the epithelial layer, it's very comparable for human, rabbit, and pig. But on the other hand, dog, pig, and human are very similar because the buccal mucosa doesn't have keratin. The sublingual mucosa of rabbit is non-keratinized. So there's pros and cons to each model clearly, but if you weigh the benefits, clearly one leans towards rabbit and dog for obvious reasons in terms of ease of performing these studies. And I'll give you examples now of how we have used both of these models to develop products that are approved and on the market. So a rabbit model here, the rabbit's oral cavity histologically similar to humans. The thickness is about 600 micrometers, so there's enough surface area for us to apply the test article. And so what you see here is an example of a drug which is a dopamine agonist. It's used for Parkinson's disease. It causes a lot of emesis, therefore less desirable to use a dog model. And this was successfully accomplished with an animal model where the injectable version has been replaced with a film strip. So this is a very, very exciting series of studies that we were involved in over the course of a year or longer, where we tested various prototype films, again, the criticality of knowing the anesthesia regimen, to ensure that there is proper administration and exposure of the test article without compromising the PK becomes, really speaks to the experience and expertise of the in vivo team here. So the application of the strip, the procedures following this, and so on all become very, very important aspects to ensure reproducibility of the results and reliability of the results in taking this formulation or product forward. So this really ended up with a PK profile that compared very favorably to the injected test article with a very good Tmax, very rapid Tmax and tolerability, showing that this is a very much easier way to treat, this type of motor fluctuations or off episodes in Parkinson's disease. So again, like I said, this is an approved product now on the market. Another very good example, this is now using a dog model, a dog in vivo model, where the sponsor was looking to alter the indication of their drug as a way to extend the patent So this is something, again, where animal models have a tremendous, provide a tremendous value, which is to help to repurpose products for patent indication. So for the new indication, they wanted the drug to be absorbed more quickly, but to be also cleared more quickly. And initial porcine buccal tissue permeability studies were performed to determine if the buccal route could be used as this alternative route and whether there were excipients that could help enhance absorption. And upon establishing that buccal was feasible and there was an excipient, menthol in this case, that caused a significant increase in absorption, we progressed to the in vivo PK studies. And this was really quite a tremendous accomplishment in terms of the number of animals that were used on a study like this, considering the complexity of the steps that were involved, because a device had to be used that was placed in the buccal compartment to localize the formulation while the dog was anesthetized. And after the recovery, the dogs had to be given an oral gavage of a urine acidifying agent, because the compound is excreted through the urine, and ionization would decrease its chances of being reabsorbed. So there were multiple complex aspects to this in vivo study, which was accomplished successfully with the endpoints, which is the decrease in Tmax, increase in Cmax, and reduction in the half-life of the compound, which is what was being sought to be achieved, was really sort of what is achieved here. And again, approved product in the market, and again, it highlights the ability of the animal model to be very reproducible and predictive. So moving on to another a completely different area, much more complex, which is the eye and ophthalmic studies, and probably an area where animal data really surpasses human data. How much human data is out there because of the limitations that we can do with human testing in this realm. So really, the regulators, the sponsors, the patients, everyone is relying on animal data to demonstrate efficacy, to demonstrate safety. And so there's a lot of work that has been done to show, and certainly interspecies comparison highlights why rabbit is one of the most well-studied, well-used models for ophthalmic evaluations. Its anterior chamber parameters are very comparable. It has a large sort of cornea and lens, and therefore an aqueous humor volume, which makes it very amenable for a lot of ocular testing. On the posterior side, the vitreous volume is not as much as humans, but it's still a very cost-effective model compared to non-human primate, in which a very, very large body of ocular data has been generated. So I'm going to show you again some more reasons, factors for why the rabbit correlates very well with human. Here's a list of important factors. When we think tear volume, you think tear turnover rate, the osmolality of the tears, the pH of the lacrimal fluids, the corneal thickness, which is very important when we think of penetration for a topical drop, the surface area that is available, the aqueous humor volume, and turnover, as I mentioned. So a lot of very good reasons to choose rabbit, and why there is such a vast amount of data using this model for ophthalmic drugs for a variety of indications. So in the next two slides, I'll show you some very interesting data, because rabbit as a model has been historically used in the development of a number of innovator or brand products, ophthalmic drugs that are out on the market. But what you're going to see here is an example of a reference versus a test comparison for the development of a complex ophthalmic suspension here, where the human study is so prohibitively challenging, and what's more, there's a high probability of failure at the end of an expensive and large human study. So this is actually prompted by the European regulatory bodies, where they have a more progressive view in terms of accepting animal data for comparisons for equivalence. So the rabbits were used. The eyes are, of course, similar in size and shape to humans, as I mentioned. The compound in question was an inhibitor of carbonic anhydrase, which is responsible for production of bicarbonate ions and is, a class of drugs that is used for glaucoma. So basically, what the outcome here was, we were not trying to look at a PD endpoint like reduction in intraocular pressure. That I'll show you in my next slide. In this particular study, we were really looking only at the distribution of the compound in the different ocular compartments when dosed with the reference versus the test. So basically, we measured the test article following dosing with each of these products, the reference products as well as the test product, and we measured the active ingredient in selected ocular tissue. And so measuring the concentration of the compound in aqueous humor shows us the levels of the test article that's passed into the anterior chamber, and measuring the concentration in the iris ciliary body shows us how much has actually reached the tissue, which is the site of the PD action. So there's a lot of value here which enabled us to ultimately select a pivotal formulation that the sponsor then took into an actual human study very successfully. The other aspect of this is utilizing animals now for a PD endpoint. Again, in a very interesting example here, we are again trying to establish equivalence between a reference article and a test formulation that is being a generic that is being developed by looking at the efficacy and safety of this sponsor's test formulation in reducing intraocular pressure after topical administration in New Zealand white rabbits. So we actually, in this case, induced a hypertensive condition, and this was steroid-induced, and then we began to administer both the test and reference formulation topically 3 times a day over a certain time period of 10 days. And then we measured IOP. And this is probably a very, very good example to show how critical it is that you pay attention to all the details when you come to a sensitive endpoint like IOP measurements, because the acclimation of the animals to the IOP measurement, taking into account the cycle, the light and dark cycle, taking into account having a dedicated researcher who is performing these measurements, these endpoints, these become very critical in being able to obtain reproducible data from a study such as this. But what we were able to show is that there was no significant difference. We had enough animals on the study. The power was sufficient to give us very strong statistical basis to show that there was no difference in IOP reduction between the reference and the test. So we have actually gathered a very, very large database of data from the rabbit for all different classes. This is dorzolamide, which is a topical carbonic anhydrase inhibitor, but we have this data for every class of glaucoma drug that is on the market, where we have the response in rabbit and we have the human response to be able to show the IV-IVC here in a sense between a preclinical model and a human condition, and then really establish that the animal model can be very predictive of human outcomes. So I'm almost wrapping up here to show you a few more highlights or utilization of preclinical models for areas outside of some oral and intranasal, intraoral, and ocular. Here's the utilization of the porcine model now because of its similarities with human for a variety of fields, whether it's for cardiovascular applications, a lot in dermatology, because the cutaneous architecture of the pig is so similar to human, and then in reconstructive and regenerative therapies as well. So I'll show you an example of a dermal model that we have developed, which is a unique application, which is a hypertrophic scar model where red Duroc pigs exhibit many of the features of hypertrophic scarring that are observed in humans. So truly, there is no other model where you can really observe this phenomenon which mimics the human disease etiology. And so utilization of this red Duroc models in a study that looked at the topical delivery of stem cells on wounds that were created has really enabled the development of a very unique therapy, which is now, again, progressing very well. So you create the wounds and apply the treatment, and you're looking at planometric measurements. You're looking at histology, obviously, which is the gold standard. But again, it is a model that has been developed and which is now, validated to a point where it can generate reproducible and robust data. So again, more examples to show the application, of course, of the swine model or the porcine model in translational research and surgical models used as an alternative to the dog and the non-human primate in a preclinical tox setting. So there's a number of different advantages to the porcine model in terms of the anatomy and the physiology involving the cardiovascular, urinary, digestive systems, and so on. So again, a lot of clinical implementation of innovative diagnostic and therapeutic technologies really depend, in our experience, working with many, many devices, really depends on the successful development of these types of large animal models that allow for the preclinical evaluation of this technology. So in closing, I want to highlight an example for those of you who are not familiar with the world of biosimilars. The FDA published a draft guidance in 2012, and it's very interesting because they emphasize the importance of analytical studies for biological products to show similarity to a reference product, but they emphasize animal studies that include assessment of toxicity and other endpoints also to show the safety or purity or potency of the test compared to the reference. So this is a landmark a poster that came out which showed how efficacy was demonstrated between a generic and the brand Copaxone in an animal model of multiple sclerosis. This generic is now approved, but it really highlights, I think, the value that animal or preclinical models are going to play increasingly in the future in an area very different from what we have seen before, which is the development of high-quality generics. So in conclusion, I would like to leave you with this thought that it's very important that we utilize these preclinical models with a lot of intelligent design. We don't rush into a study without thinking through about the choice of the animal model, about the power that we want for the study, the type of endpoints that need to be measured, the components of the formulation that are being used to deliver the test article, the bioanalytical platform that will generate the data, the results, and finally the interpretation of that data, of those results, to really utilize what are probably the most valuable models in research, these intact animal models, and being able to make these types of high-resolution measurements, especially if we're able to induce a disease state in these animals, is really going to provide a lot of information about the underlying biology that's going to help us elucidate for a new drug or compare for an existing drug efficacy and safety in a robust and reproducible manner. Thank you very much.
Well, those were great presentations. You might see a poll that pops up right now, so please answer. I'd just like to once again thank you both, Vatsala and Stacy, for such a terrific presentation. And before we get started on the question and answer session, I would like to remind our audience that this educational webinar offers free continuing education credits. So after the webinar is over, to get your CE credits, please click on the CE button located at the bottom left-hand corner of your webpage. And as a quick reminder of how to submit questions, you can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. So the first question that we have is from Lillian, and she asks, Could the intellectual property concerns make the model not reproducible?
Thank you. So this is Stacy, and I will go ahead and answer this one. Yes, IP concerns actually can make a model, can decrease the reproducibility of a model. Due to IP concerns, not all of the necessary details for reproducibility may be published, and companies may not be willing to share that information even outside of publication. That has happened before in the pharmaceutical industries. I've personally experienced it when I used to work at CROs. That's something that I've seen a few initiatives over the years with pharmaceutical companies in particular, promising to provide more information about what they're doing to help with that issue. But right now, the bigger issues seem to be with statistical underpowering in academia, as well as making sure animal-related details are published.
Great, thank you, Stacy, for that. We have another question, and our audience member asks, how do I choose the right animal model to test my therapeutic or device? Vatsala, do you want to answer that question?
Yeah, I'm happy to answer that, having shown you some examples now of different sort of therapeutic areas and endpoints where we've utilized different animal models. And it really is a good question, but a broad question, because I think it starts with being able to sort of understand what is the therapeutic indication and what endpoints you're looking for, and whether this is feasible within the scope of the animal model that is being utilized. Then I think the anatomy and the physiology, those aspects, those parameters have to be taken into account, as also other important factors such as the immune response and other parameters that are related to the animal. If you're looking at an intraoral product, rodent model versus a rabbit model, the size of the oral cavity, the saliva, the pH of the oral cavity, the thickness of the epithelial layer, the presence of keratin or absence of, all these become very important. So it really depends on whether you're looking at a specific dosing route or you're looking at a specific disease indication, whether that can be induced or those symptoms can be recapitulated in the animal model.
Great, thank you for that. We have time for one last question, and our audience member asks, what are some of the key factors that should be considered when developing a protocol for a preclinical study?
I think statistics, the sample size, this was an important point that Stacy brought up, and with reference to an IACUC view on this, but really sample size, being able to calculate this, making certain important assumptions which can be defended and to support the sample size that you're using, the study, the statistical methods that you're planning to use. I think the randomization, more and more, I think we're sort of starting to see the benefit of almost applying some of the parameters that are used for clinical studies, even in a preclinical setting. So randomizing the animals to groups so that you minimize bias, making sure the animals are acclimated, perhaps using any type of blinding methods so that you're not having any bias again. Certainly looking at the PD and the PK considerations, the administration of the dose, the time points, and any adverse effects that can impact the animals and thereby the conclusions of the study. I think all this needs to be taken into account when you're developing a protocol.
Great points. I would like once again to thank both you and Stacy for your presentations. Do you guys have any final comments? I'll start with Stacy. Do you have any final comments?
No, I think I guess yes. What Vatsala mentioned is true with regards to looking at model selection as well as study design for preclinical studies. There are also some regulatory guidelines that have to be met, again, as Vatsala mentioned. But the more reproducible that we make specifically preclinical studies, the kinds that Vatsala described, the more translatability we will get for human research. And so in line with the ethical arguments for reproducibility, it works very well.
Yes, I'd like to just say that, I hope these web presentations have given the audience a lot of food for thought. I certainly, it made me, it was very thought-provoking, the points that Stacy made, and I think we have to keep this very much in our mind and for consideration when we are designing these studies, keeping the use of animals to ensure that it is really giving us valuable data that can enable development of drugs and devices. So thank you very much for listening to us this morning.
Well, great. Today's webcast will be available for on-demand viewing through August of 2016. As an audience, you will receive an email from LabRoots alerting you when this webcast will be available for replay. And we invite you to forward that announcement to your colleagues who may have missed today's lab event. That's all the things we have for today, and we'll hope to see you again next time. Bye-bye.
Bye-bye.